Muy bien, empiezo entonces. Muchas
Dale,
gracias.
yo les cuento, bueno, ya te conocen muchos de los que están acá, pero
Dale,
les cuento
sí.
que hoy tenemos a Gabriela, que, bueno, a la UMA suele, digamos, costarnos traer argentinos que están viviendo afuera, porque son congresos donde la sesión de estadística es chiquita, entonces aprovechando la virtualidad, invitamos a Gabriela, que amablemente aceptó estar acá con nosotros. Gabriela es argentina y está en Canadá, en la Universidad de British Columbia, y trabaja en robustez, alta dimensión, ¿no? Y aplicaciones. Hace cosas súper interesantes. Así que es un placer tenerla hoy con nosotros.
Muchas gracias, Daniela, y gracias también a Paola por la invitación. La verdad es que es un gusto participar de la reunión y si bien estoy en un momento del term recontra complejo porque tengo que enseñar 3 cursos nuevos y aplicaciones y cosas, me encantó poder participar y poder ver a mis colegas allá en Argentina también, así que muchas gracias por invitarme. Lo único es que soy muy argentina, pero la presentación la voy a dar en inglés, como les dije, porque hay mucho lenguaje técnico que básicamente lo manejo mejor en inglés que en español. Y sí, trabajo en varias cosas, pero en general trabajo en la intersección de robustez, high dimensional y aplicaciones a estadística genómica. Y hoy lo que voy a presentar está en la intersección de high dimensional omics data y una nueva parte que se llama instrumental variables, que es una técnica para resolver problemas de causalidad. Ok, de ahora en más no voy a mezclar más idiomas. Oops. Okay, so with new technologies and with new ways of measuring many features in the human organism, there has been a big effort to try to personalize medicine and medicine initiatives, basically. And so that is the motivation of my methodological work in this talk, although I'm not going to make a lot of medical interpretations because I'm not really the doctor here. But my methodological work has been inspired in these new initiatives. So we already know that there are biomarkers out there that can be used to detect or to diagnose different diseases. For example, we can measure serum LDL to know if we are having cholesterol or as a surrogate for many heart diseases. We can also measure troponin as a biomarker to myocardial infarctions, or creatinine is a very common biomarker to identify kidney diseases. However, now there are many of these markers or many of these features that we can measure from different tissues or from cells or from fluids like urine or blood or saliva. But among all those that we measure, and sometimes the dimensions are huge, how many are really markers? How many tell us, or which one tell us what is happening in the organism? And those are not too many. So we're in a very, very sparse space here. So in particular, in this study, we have looked at cancer data proteomics, trying to identify markers. And there has been previous studies that have noticed an association between proteins and prostate cancer Gleason score, which is a score that has been used to know the progression or the aggression of prostate cancer. However, many of these identified relationships that may improve the diagnosis of aggressiveness might not be causal, and that's where my work comes in. So, for example, we can think of a very simple way of modeling that relation. It can be through a linear model. So I have here a very naive linear model where yi will be the response. In this case, it will be the Gleason score, and xi's can be different variables that we are going to use to predict that score. So in my case, those variables are going to be proteins that we measure in blood. We can also put demographic variables, and it can also be genomics variables and other medical variables. But the main problem here is that in observational omics studies, there are usually many confounding factors. Environment can alter the expression of the proteins and the expression of the genes. There can be measurement errors, and there are. And there is this also problem called reverse causality problem, where we don't know if it is the protein causing a change in the Gleason score, or if it's the Gleason score which changes the expression of the protein. And it can be both things. Now, statistically, what is happening here is that the covariate xj, for simplicity, I'm just saying that there's only one with problems here, will be correlated with the error term. And if in a linear regression, the x is correlated with the error term, the classical estimators are going to be biased and inconsistent. So a proposed solution to that problem has been instrumental variables estimators. And because I don't know if you're familiar with instrumental variables estimators, I'm going to make a quick review of them. The main idea of instrumental variables estimators is to try to separate from the x that has problems a part that is uncorrelated with the error term from the part that is correlated with the error term. If we can separate these two, then we can use the uncorrelated part and be back in the usual classical settings. So to do that, we use what is called an instrumental variable. So this is an additional variable that we need in our data set. And the difficulty sometimes comes of what is that variable? Where can we find that variable? Has it been measured? But to succeed in this division of uncorrelated and correlated parts, there are some conditions that the instrumental variable needs to satisfy. So the instrumental variable needs to be associated with the variable of interest, because otherwise we can't really do anything about it if they are not related at all. The instrumental variable does not need to affect the y except through the relationship in X, otherwise these would never be able to be identified. And nothing else should be affecting W and Y at the same time. So we don't want any what we call backdoor paths here. So if we are able to do that, what we can do then is to project our problematic X on the space of this saver W. Because everything that is projected in W, we know now that is not, because of these conditions, will not be correlated with the error term. So basically, this projection into the instrumental variable space will be the uncorrelated part. And all the rest will be what remains being correlated. So the idea, of course, is that we can use least squares to get that projection and to get an optimal projection there, and that part will not be correlated with the error term. And if we do that, we get an estimator called this two-stage least squares estimator. Why two-stage? Because in the first stage, we project and we get X hat. And in the second stage, we run another least squares estimator again to get the beta hats that we were looking for. So this estimator called two-stage least squares estimator, it can be proved that it will be consistent. It's not new at all. It has been proposed in the 30s by Philip Wright in the context of economics, although it has been arguably attributed to his son, who was a genetic statistician called Sewell Wright. Now, it has appeared in the 2000s in many biomedical studies, and I'm giving some references here, in particular in epidemiological studies. Now, the way that they appear in the epidemiological studies usually have a different name. They are called Mendelian randomizations. Why the name? What Mendelian randomization studies? At the end of the day, they are using instrumental variables estimators. Well, the idea is that, and I'm kind of showing you a little bit of pictures here, is that we are looking for a protein that can explain what is happening in the tissue in this case, or in the Gleason score. So we are looking for this association, but we know that this association can be confounded, and we know that we need help for this. So what can we use? There's a dogma in biology called the central dogma of biology that says that the protein is responding to the information that they get from genes, and the genes are responding to our genetic information. And that genetic information, if we have a genetic variant, it has been randomly assigned during meiosis. So this is like a natural randomized experiment. If we can use those genetic variants that we know at the end of the day will be related to the proteins and will not be related to prostate cancer unless through this path. So people have realized that they can use those genetic variants to try to identify causal effects in biology, and they call them Mendelian randomization studies. So then after doing that first stage, what we do is that we use the predicted protein values to consistently estimate the relation between this relationship that we have here in blue. So that's the main idea of Mendelian randomizations. Now, what is the problem with that? That appears in the about 2000s, as I showed you before. Well, the main problem is that we don't have only one protein, and we don't have only one gene, and the technologies has evolved, and we are measuring thousands and thousands of genes and proteins and genetic variants. So for example, in the human chromosome with 23 pairs, there are about 3 billion pairs of DNA information that has been encoded into 20 to 25,000 of genes that then give information to more than 100,000 proteins. So we are also in a huge space. So the Mendelian randomization studies of the 2000s were only one gene and only one genetic variant, not this huge space. But now we have more data, so can we still do something here? And that is why the title of my talk, Upscaling Mendelian randomization. So that is basically the goal. What can we use in those cases? Oops, sorry. So finding valid instruments is difficult. We are measuring too many, and sometimes the signal is very low as well. The instruments sometimes are discovered to violate one of those conditions, and if the instruments violate the conditions, the instruments are variables estimators do not really make to their promise. They are still as bad as the two-stately square estimators. And we have many proteins as well. It's not that we have many instruments only, we also have many proteins. So the literature today is trying to upscale all these. So some people have said, okay, let's suppose we have only one protein and let's look at the problem of many instruments, or let's look at only one instrument but many proteins. We are trying to put all the problems together to solve them. So can we do that? Can we design something that will work in high dimensional settings, and even when we have more instrumental variables, for example, that subjects? And so here we call lasso to the rescue. Like, this smells a lot like variable selection. So can we use a technique like lasso or something similar to select variables? So we have used something similar to lasso, one called post lasso. So Bellioni, it's econometrician, has proposed post lasso, but in a lower dimensional, having only one covariate and many instrumental variables, as I was saying before, and not in the context of Mendelian randomization. So we are adapting his estimator to this new problem that we are facing. Post lasso means that we use lasso to select variables, but then we use something else to make the estimations of the coefficients. So for example, we can use least squares on the selected sets given by lasso. So we use, with this 3-stage instrumental variable estimator that we designed, what we are doing is we are regressing the proteins on the genetic variants. Then we are regressing the clinical outcome, for example, the Gleason score on the predicted proteins. And we are also using it to test violations of the instruments, because if the instruments are not good, we are not going to have a good estimator. And because of that, we call it a Thrive estimator. So Thrive comes from 3 times robust instrumental variables estimator. This is not robustness to outliers. This is basically robustness to violations of the instruments. Okay, so I'm going to, I know we don't have a lot of time, so I'm going to present you the results of a simulation study, and I'm not giving you all the details, so feel free to ask me questions after of how I generated these variables. But the important part perhaps here is to know that we have 10 endogenous proteins. So by endogenous, it means that they are correlated with the error term. So we have 10 proteins with problems. Now of those 10 proteins, 5 are relevant to, let's say, to predict the Gleason score. The other ones are just noise. Of course, in real life, we have many of these. It's not just 10. And then we look at what happened when we increase the number of instrumental variables. So we are under a low setting here for the covariates, but we are increasing the dimensionality of the instrumental variables. And in our simulation, we always have 1 relevant instrumental variables for each protein. The other ones are just noise. The sample size is 500. And we have more simulation studies in our paper where we played a lot with all these parameters. What I'm showing you in this graph is the median absolute deviation of the coefficients, and we are comparing many estimators. Well, one estimator is the least squares. The least squares, it's really not using any instrumental variables. So it's one that it really does not address the confounding factors, or it doesn't address the endogeneity. It doesn't address that the X is correlated with the error term. We are also using plain lasso, again, without any instrumental variables. And those 2 are the ones here, are the ones that has the highest MAD in this case. We are also comparing it with the 2-stage least squares. So the 2-stage least squares will not make any selection. It will use all the proteins and all the instrumental variables. And you can see how it deteriorates when we have many instruments, because it's trying to use a lot of information that is really useless to make the predictions. Then we have these last three methods. Lasso, it's basically using lasso in the 2 stages, or plasso, which is the post-lasso in the 2 stages. And we added also using post-lasso to identify instruments, but then least squares for the proteins. Because the main problem in this setting is the instruments that are increasing, but we are still in a very low dimensional setting for the protein. So you can see that of all these post-lasso is the one that has a very good performance, at least in this low dimensional setting. We also look at the variable selection properties, because that's also very important, because if we select the right proteins, those are the ones that eventually can lead to an improvement in care. So there we compare lasso and plasso, and we can see also how this plasso estimator is doing very well in terms of selecting important proteins. In this plot, the difference between the grid and the non-grid is really a technicality that I don't have time to address, but I would say that in our paper, one of the things we are proposing is how we tune this selection. And so this one is showing that tuning it with an appropriate grid helps a lot when we are selecting variables. The other setting, we increase the proteins to 50. And when we increase the protein to 50, we can also see that the 2-stage least squares estimators with variable selection are doing well and better than not addressing anything at all. And then in terms of identifying the relevant proteins, also Plazo is still a leading estimator there. LASU without anything, it does. This one is really not identifying anything. That's why it's not doing well at all. Okay, so I think I'm almost done here. What I'm not going to present anything on the proteomics. We have used real data from the genetic atlas that is available online, and we requested the genetic information. It's a data you need to request. It's not available publicly. So because of those privacy issues, I decided not to include any of the results of the prostate cancer study. We are in the process of finishing to write our paper at this point, and all the simulations has been done, and the application to prostate cancer has been done. But what I can say is that because in the application we identified 2 proteins that sounds very interesting, they have interesting biology, and they show good properties overall, statistical properties in terms of trying to identify causal relationships with prostate cancer. So having said that, I want to thank Joe and David. They were my students. They both graduated now. Joe is doing a postdoc in Canada, and David has a tenure track position in George Mason at the moment. And I want to thank them both for all the contributions to this project, and we hope to finish soon our paper. And with that, I will conclude. Basically, what I told you before, recent advances in omics technology has simulated a large number of biomarkers and discovery studies. This talk in particular, we focus on addressing problems, and sorry for the typo there, in observational studies to identify causal effects in omics data. We propose a new estimator called Thrive, and I haven't told you all the details on how we made Thrive. It's based on post-LASU estimation, but it has been customized to the problem we are trying to solve here, which is Mendelian randomization studies. Thrive integrates genetic and proteomics information, but it can integrate genetic and genomic information or other basically omics data available. And we have applied for a study of prostate cancer progression, although I haven't shown you any of those results. Okay, so having said that, I want to thank you for your attention. This is a picture of Vancouver for those of you that have not came here, and I hope that COVID restrictions get released soon and you can all come and visit. Thank you. So I think we have some time for questions, if you have any questions.
Muchas gracias, Gabriela, por la charla. No sé si tienen alguna pregunta. Muy linda charla.
Sí, Graciela. Y me podés preguntar en español, ya podemos, no hay problema.
Y la pregunta es, ustedes usan LASU, ¿está bien? Ahora, LASU no siempre hace selección de variables, tiene que cumplir ciertas propiedades, digamos. Mientras que SCAD y MCP sí hace selección de variables. ¿Probaron usar SCAD, MCP o hay problema? O sea, es más compleja la parte numérica con SCAD y MCP porque, digamos, la SU es convexa y las otras son cóncavas, digamos, ¿no?
Sí, no probamos, está en nuestra to-do list para seguir adelante. Pero lo que sí probamos fue usar ElasticNet, que no tiene las mismas limitaciones que la SU, y se comportaba muy parecido. Por eso que no quise poner muchas cosas acá. Pero sí queremos ver qué pasa con las non-convex penalties. Eso sí nos interesa ver qué pasa ahí.
Sí, porque suelen dar mejores resultados en general.
Sí. Y aparte, otro problema que tenemos acá es que en este mundo de omics, todo están relacionados. Los genes, las proteínas, hay mucha correlación, hay muchos grupos. que los biólogos llaman pathways. Entonces la SU va a seleccionar también uno de esos grupos.
De esos grupos, sí.
Y podemos estar perdiendo mucha información también por hacer eso. Por eso que probamos con ElasticNet, pero los resultados eran muy parecidos. Entonces decidimos cerrar por acá y tratar de terminar el paper y después pasar a otras penalties.
Claro, es que el problema de la correlación en grupos es un problema serio en selección de variables, digamos.
Sí, sí. Y
pero
mi ex alumno David,
sí.
Sí, David está, sí, vos lo conociste, está trabajando.
Yo lo conocí, sí.
En usar Adaptive Lasso,
Sí,
otras,
sí, que es mejor.
Otras penalties, sí. Y la parte que está faltando acá, que espero poder empezar a trabajar con una nueva alumna que tengo este año, es agregar la parte de robustez realmente. Que no es por ser fanática en robustez, pero realmente estos datasets están repletos de outliers. Claro. Y hay veces que entonces terminas encontrando una proteína solamente porque estaba tan llena de outliers que lo que estás haciendo es detectando outliers nada más. Entonces faltaría agregar el componente robusto, en el sentido robusto outliers. Con Rubén yo trabajé en un paper de Robust Instrumental Variables, pero no tiene la parte de High Dimensional. Entonces,
claro, acá los outliers son cell-wise outliers, además, supongo.
Y acá los outliers van a ser cell-wise outliers, muy probablemente, sí. Sí, no vamos a perder un montón de información, sí. Sí, así que bueno, quedan un montón de cosas abiertas y eso es lo lindo, pero es muy difícil probar cosas, es muy difícil encontrar resultados consistentes, no en el sentido matemático, como que uno tiene una hipótesis y lo ves y en algunas cosas, algunas simulaciones funcionan, en otras no, y es muy difícil encontrar una historia que cierre por todas partes. Pero es muy interesante el tema también, así que aquí sigo.
Muy lindo, la verdad que
gracias.
Muy, muy lindo.
Gracias, Graciela. No sé si hay preguntas en el chat, veo que hay...
Agradecimientos hay.
Agradecimientos hay.
Muchas gracias. De Alejandra, gracias Alejandra. Hace mucho que no nos vemos, sí. Espero poder ir a Argentina pronto. La verdad es que el COVID me mató. Siempre estar acá o en Estados Unidos estuvo bien siempre que pude viajar y nunca me imaginé que me iba a pasar esto de quedar atrapada sin salida. Así que extraño mucho, obviamente, a mi familia, a mis amigos, Argentina, todo. Así que voy a tratar de ir en diciembre, espero manteniendo los dedos cruzados para que no explote nada y podamos ir a ver. Así que si voy les aviso y nos vemos.
Sí, te recibiremos con gusto.
Muchas gracias. Y cuando quieran pueden venir a Vancouver. También si tienen alumnos que quieran mandar para acá de visita o para trabajar en algo, me avisan.
Genial. Bueno. Muchas gracias, Gabriela.
Muchas gracias, Gabriela.
No, gracias a ustedes. Me alegro que no me pasé mucho del tiempo. Tenía miedo que fuera muy largo.
Espectacular.
Estuviste muy bien.
Muy bien.
Bárbaro,
muchas gracias, Gabriela.
Hasta luego.
Gracias,
Chao.
hasta luego. Chao, chao.
Gracias, hasta luego.